The Gram stain is named after its inventor, Hans Christian Gram. As a Danish botanist and teacher of pharmacology, he devised this important method of differentiating bacteria in 1884 that is still in common usage today. The Gram stain is used to distinguish between gram-positive and gram-negative cells. It is the most commonly used method to observe bacteria. The Gram stain provides two important features: the Gram reaction, or color of the cell, and the cell morphology, the shape of the cell. Bacterial cells can be spherical, rod, or corkscrew in shape, and nearly all clinically significant bacteria can be differentiated into these three groups. Knowing which type is causing an infection is critical to patient care. The Gram stain differentiates cells based upon differences in the structure of the cell wall. The cell wall is made up of peptidoglycan, which consists of sugars and amino acids that form a layer around the plasma membrane of most bacteria. Bacteria that stain dark blue-purple are called gram-positive. They have a thick peptidoglycan layer that retains the primary dye. Conversely, bacteria that stain pink to red are called gram-negative. They have a thin peptidoglycan layer that loses the primary dye when flooded with alcohol. The Gram stain begins by adding 1 drop of sterile saline or deionized water onto the center of the slide. Then take a sterile loop and touch the edge of an isolated colony on the Petri plate. Place the loop into the drop on the slide and swirl it around to distribute the cells evenly, so the cells are 1 layer thick. Using cells from a single colony is important, as this should represent only one cell type rather than a mixture of cells. Ideally, the culture should be between 18 and 24 hours old, because older cells may show variable results. Alternatively, you can use a collection swab from a clinical specimen or environmental sample. Do not heat the slide. Wait for the sample to air dry. Bacterial cells are then fixed to the slide using methanol. Fixing cells is important, as it prevents the cells from washing off during the staining process. Methanol fixation is recommended rather than heat fixation, as methanol preserves the cell's morphology. Heating the slide will cause cell distortion, could increase cell debris, and may cause erroneous Gram reactions. Place or hold the slide over a paper towel, and flood the slide with absolute methanol for 2 minutes. Alternatively, you may dip the slide into a Copeland jar filled with methanol. Once 2 minutes have passed, tilt the slide and drain off the excess methanol, and let the slide air dry. Do not wipe or blot the slide, as this can remove cells. Do not heat the slide. Cover the slide with the primary stain, crystal violet, for 1 minute. Crystal violet is a dark blue to purple dye that stains all cells. Rinse the slide with deionized or tap water to remove excess crystal violet. Flood the slide with Gram's iodine for 1 minute. The negatively charged iodine molecules act as a mordant and link to the positively charged crystal violet. This traps the crystal violet in the peptidoglycan layer. Once 1 minute is up, rinse off the Gram's iodine with deionized or tap water. Tilt the slide at an angle over the sink or tray and add the decolorizer dropwise until the violet color stops running. When the solution runs clear, immediately rinse the slide with deionized or tap water to neutralize the decolorizer. The decolorizer is composed of a ratio of alcohol to acetone. The ratio is normally a personal preference, with faster decolorizers containing higher concentrations of acetone and slower ones containing higher concentrations of alcohol. When decolorizer is added to the smear, lipids are extracted from the cell wall of Gram-negative bacteria. Lipid extraction causes an increase in cell wall permeability, and the cell becomes leaky, resulting in the loss of the purple dye mordant complex from the thin peptidoglycan layer. In contrast, the effect of decolorizer on Gram-positive bacteria is dehydration. This decreases the cell wall permeability, causing Gram-positive cells to retain the crystal violet Gram's iodine complex. Note that a thick smear will require more decolorization than a thin smear. Be sure to stop the decolorization process immediately after the blue dye stops running off the slide. Beware. Over-decolorization can result in false Gram-negative results. Beware. Under-decolorization can result in false Gram-positive results. In essence, Gram-positive cells will be blue-purple, and Gram-negative cells will be colorless after decolorization. The final step utilizes a counterstain of a different color to differentiate gram-negative from gram-positive cells. Cover the slide with safranin or carbofuscin for 1 minute. The gram-negative bacteria will absorb this dye, causing them to stain pink to red. There will be no effect to the gram-positive cells since they retain the initial crystal violet stain. Rinse the slide with deionized or tap water. Do not wash the slide excessively. Allow the slide to air dry by tilting it onto a paper towel or over a sink and allowing it to air dry. Alternatively, gently dry the slide by blotting it using a lint-free bibulous paper. Do not use a wiping motion, for this can remove the smear. To summarize, crystal violet exposure, 30 to 60 seconds. Iodine exposure, 30 to 60 seconds. Acetone alcohol decolorizer, only enough time to rinse away the blue stain. Safranin exposure, 30 to 60 seconds. The slide can now be viewed under the microscope. First, focus on the image using the high dry objective lens marked 40x. Then, without removing the slide, switch to the high power oil immersion objective lens that is marked 100x. This will result in an overall magnification of 1000x. Here you see two organisms routinely used for quality inspection at Hardy Diagnostics. The purple gram-positive cocci are Staph aureus. The pink gram-negative rods are E. coli. Hardy Diagnostics offers an improved formulation of GramStain called GramStain Advanced. This kit produces superior stain results with bright, vivid colors, even for bacteria that are difficult to stain. To test the quality of your stain and staining technique, Hardy Diagnostics offers quality control slides, the Q-Slide Gram, that are pre-inoculated and methanol fixed with gram-negative and positive bacteria. Thank you for watching this GramStain demonstration. Hardy Diagnostics is your complete one-stop shop for all your microbiology supplies. Give us a call and experience our culture of service.